Assalamualaikum, how are you? My name is Manzafar Pracha. Recently, I made an illustration on prime editing. Uh, this is the presentation of that illustration. Based on this information, let me tell you that this presentation could be of much help to you, could easily be understood if you open that illustration while listening to this presentation. The link to that illustration is presented in the description. Nevertheless, prime editing is a gene editing technology that has a huge potential with applications in plants. Prime editing has two basic components. One is prime editor, and the second is prime editing guide PEG-RNA. Prime editor has further two components. One is Cas9. Nick is abbreviated as Cas9N. It is a mutant protein that can cut only one strand of DNA. And the second component of prime editor is reverse transcriptase. It is an RNA dependent DNA polymerase, an enzyme that performs the required editing. On the other hand, PEG-RNA, prime editing guide RNA, has three components. One is gRNA target sequence, second is primer binding sequence, and the third is reverse transcriptase template with desired edited RNA sequence. So prime editing has two components: prime editor and prime editing guide RNA. Uh, Concerning its process, there are six basic steps in its process. The first step is the complexation of prime editor with prime editing guide RNA. In the second step, the prime editing guide RNA guides and takes the complex to the target DNA, where target DNA binds with the complex. In this process, Cas9N nicks a strand of DNA. In the third process, Cas9N cuts DNA on one strand. In the fourth process, there is incorporation of desired sequence into target DNA by reverse transcription of RNA. In the fifth process, there is incorporation of edited strand with cleavage of original DNA with cellular endonuclease enzyme, with the help of cellular endonuclease enzyme. Eventually, in the sixth process, there is repair of DNA, repairing of unedited strand according to newly edited sequence, so that both strands of DNA comes with desired edit. So this is the process of prime editing. As it may sound, it is a complicated process. So I told previously that this presentation could be of sufficient information only when you open that illustration along with this presentation. Before going further, before ending the presentation, I want to present a little summary in German language. Hallo, ich bin Manzafar Pracha. Das ist Zusammenfassung in deutscher Sprache für Prime Editing. Diese Gene-Editing-Technologie bietet ein großes Potenzial für Anwendungen in Pflanzen. Bei dieser Technologie komplexiert ein Prime Editor mit der Prime Editing Guide RNA, PEG-RNA, und bindet an die DNA, in der die gewünschte Sequenz hinzugefügt wird. Danke. In the last, I want to say that if you like this presentation, or if you like me to present such types of presentations in the future, or to make them better, you can support me on Patreon. Thank you.